Now we are at the point of looking at Chapter 6 for our Bio 100 class, where we look at cellular respiration and ways in which cells gain energy from their food. One of the most critical components to either aerobic or anaerobic respiration is glycolysis. And now please keep in mind that aerobic respiration occurs in the presence of oxygen and anaerobic respiration occurs in the absence of oxygen. Obviously, all cells first get their food in some form, and food has to be broken down first, well, for people, by chewing and digestion and other forms. Eventually this food gets broken down into simple sugars. A 6-carbon, generally our ideal, the one that we're going to look at the most, is a 6-carbon molecule that we're all familiar with called glucose. And once your food is broken down into glucose, this is where the cell takes over to actually harvest energy from that food. Now, glycolysis occurs in the cytoplasm of the cell. It does occur in all cells, and it is the precursor, it is the first step of obtaining energy from your food for both aerobic and anaerobic processes. Another thing to keep in mind is that there are many reasons that glycolysis is considered likely to be one of the first evolutionary ways for cells to obtain energy from their food, in part because it occurs in the cytoplasm and does not require any organelles. So therefore, bacteria can employ this method as well to obtain energy from molecules similar to glucose or other simple sugar. It's also likely to have occurred early in evolution, as it proceeds readily in an anaerobic environment, and early in the Earth's history there was not oxygen readily available or in the environment. It does not require organelles, so some of the first cells to exist possibly employed this method. Now we will go right into glycolysis. So glycolysis, as we talked about, happens in all cells, but in animals and plants it is just part of the process of aerobic respiration. It starts out here by breaking down your glucose and getting some ATP, but eventually those products, pyruvic acid, are brought into the mitochondria, where plant and animal cells continue with the citric acid cycle and then the electron transport chain to gain more energy from the bonds in glucose. But now we'll just look at glycolysis, because this can lead to either aerobic respiration, like this picture shows, or anaerobic respiration, which is also called fermentation. This occurs in animal cells, in muscle cells, as well as in many microorganisms. So this is an overall diagram to get you familiar with the process of glycolysis, with a key at the bottom here to show you that the gray spheres are carbon atoms. P in a yellow circle is a phosphate group, and these represent, the little minus signs in a yellow sphere represent high energy electrons. For this picture, they break glycolysis into two different steps, where the first step they call the energy investment phase, because in the first step it actually requires the use of 2 ATP molecules to break this 6-carbon molecule, glucose, into 2 different 3-carbon molecules. And when they do break it, the phosphate group, remember, when ATP provides energy to a system, it forms ADP and loses a phosphate. And here are those phosphate groups. Moving on, another phosphate comes into play. After NAD+ accepts 2 electrons from this molecule, it ends up with a phosphate on either side. For the third step, this is enzyme facilitated, but this is where 2 ATPs are generated for each molecule that glucose is broken into. The resulting products are 2 pyruvic acid molecules. So we're going to go into this a little more in depth. So the first step, first we're looking at glucose, 6-carbon molecule. We know that glucose actually has a structure more similar to this green hexagon, but for the purposes of understanding glycolysis and further processes in cellular respiration, it's going to be represented like this. The overall look at glycolysis, your input is glucose, your reactants are glucose, and your output, or your products, are 2 pyruvic acid molecules. So one glucose makes 2 pyruvic acids. and this is how we start. Remember, the first step requires ATP, so your body uses energy in order to break that glucose molecule into 2 3-carbon molecules. In the process, those extra phosphate groups are added to one end of each of those molecules. Next, this wonderful NAD+, which is critical for each step of cellular respiration as well as each step of fermentation, so you're going to become very familiar with this little guy. Remember, it's positively charged, so it can accept 1 electron, and then as a hydrogen gets added to it, it can accept another electron. So it has room for 2 electrons. What will happen to each of these 3-carbon molecules then? When they encounter NAD+, it will take 2 high-energy electrons from this molecule with a phosphate readily present that'll be attached to the other end. Now remember, for each single glucose molecule, this will happen twice. You end up with this molecule here, which is called 1,3,5-phosphoglycerate, BPG. We're not going to go into that. That's no fun. So this picture simplifies the energy investment phase, or joins the energy investment phase with the energy harvest phase. Moving on, there is one final step to get us to the product side, where 2 ADPs are required to come into play. An enzyme will facilitate the process in breaking the phosphate group off of this 3-carbon molecule and attaching it to an ADP. So this will happen for this single 3-carbon molecule twice. You get 2 ATPs. Same with this one, 2 ATPs. So let's go back and count ATPs. Right here, we're short. We've used 2 ATP molecules for 1 glucose molecule. And then off of the 3 carbons, we gain 2 ATPs here and 2 ATPs here. So essentially you generated 4, but used 2. That leaves you with a surplus of 2 ATPs. So once ATP is removed from this intermediary molecule, after we count the ATPs, just keep in mind it is an enzyme that facilitates the breaking of that phosphate group off of the 3-carbon molecule and connecting it to an ADP. Remember, an enzyme reduces the activation energy, or the energy required to add that phosphate group on to the ADP. So you are left with a TP and then a 3-carbon molecule with a phosphate, but the same enzyme will do the same thing on this end of the molecule. Therefore, bringing you to our original picture, once these 2 phosphate groups are broken off, you get 2 simple 3-carbon molecules, which are called pyruvic acid. So to quickly break it down, remember your food is broken down into simple sugars like glucose. Your cells use glycolysis. Remember, glyco sort of means sweet and lysis means to break. So you're breaking down that sugar and garnering energy for it. Glycolysis occurs in the cytoplasm. It occurs in all cells, which is part of the reason it is likely one of the earliest evolutionary methods to gain energy from sugar or other food molecules. Now, once you get your products from glycolysis, you'll use those pyruvates. If you go into cellular respiration, they will be used in the citric acid cycle, or in terms of fermentation, those 2 pyruvate molecules end up being moved into different product molecules where NAD+ can then be recycled. You should see the anaerobic lecture for that.